we are going to begin our first infection. That infection we call cholera infection. So this cholera infection in Marathi we call it as a patki disease. Here continuous diarrhea is the result of this disease. So this cholera disease is caused by a bacteria. Here we are going to study their etiology, pathogenesis, clinical features, laboratory diagnosis, epidemiology, treatment and prophylaxis of cholera. So firstly, etiology. This term etiology, it is used to describe the clinical features, the morphological, biochemical features of that pathogen. Means you can simply define this as a characters of that pathogen. It is shown in the laboratory, that we call it the etiology. Pathogenesis, it is the term which is used to describe how this pathogen enters the host, affects which parts of the host, and what type of features produces in that host. Means how this disease, cholera disease, is produced in the host, that we call as a pathogenesis. Clinical features. These clinical features, you simply define these as a symptomology. Means symptom. What symptoms are produced by this disease in the host, that we call as a clinical features. So next point is the laboratory diagnosis. How we will diagnose that person suffered from the cholera disease. Cholera disease कशामुळे होतो, कोणत्या पॅथोजनमुळे होतो, हे डायग्नोसिस करण्यासाठी काही लॅबोरेटरी टेस्ट असतात. त्या लॅबोरेटरी टेस्ट केलं की आपल्याला माहिती होतं की तो पर्सन त्या डिसीजने सफर्ड आहे का नाही. हे आपण लॅबोरेटरी डायग्नोसिस मध्ये पाहत असतो. नंतर पुढचा पार्ट जो आहे तो एपिडेमॉलॉजीचा आहे. हा एपिडेमॉलॉजीचा पार्ट जो आहे, इट इज सिम्पली डिफाइंड ॲज अ स्प्रेड ऑफ अ डिसीज इन अ कम्युनिटी. कम्युनिटीमध्ये कशा पद्धतीने, कोणत्या सोर्स, कोणत्या मोड न हा डिसीज कम्युनिटीमध्ये स्प्रेड होतो, हे सर्व आपण त्या एपिडेमॉलॉजी ह्या टायटल खाली पाहणार आहोत. आणि लास्टली ट्रीटमेंट अँड प्रोफिलॅक्सिस. याच्यामध्ये आपण अशा पद्धती काय ट्रीटमेंट करणार, म्हणजे तो कॉलरापासून तो पर्सन रिकवर होईल, त्याला आपण म्हणणार, त्याला जे काही अँटीबायोटिक्स देणार, न्यूट्रियन्स देणार, सलाईन देणार, ते सर्व ह्या ट्रीटमेंटमध्येच येणार. लास्टली द प्रोफिलॅक्सिस. कॉलरा होऊ नये याच्यासाठी आपण काय प्रिकॉशन घेऊ शकतो, what type of precaution we can use to avoid the cholera, that comes under the heading prophylaxis. So we will begin our first disease, that is cholera disease. This cholera pathogen, it belongs to a genera Vibrio, and the characteristic of this Vibrio genera, they are these bacteria which belong to this genus Vibrio, they are gram negative, rigid curved rod. When we observe this bacteria under the microscope, you are able to observe that this bacteria is a rod-shaped rod with curve in that morphology. And these bacteria belonging to this Vibrio genus, they are actively motile by means of a polar flagellum. Means they are having a single flagella. With that single flagella, they are able to move. They are able to show the movement. Okay. The next characteristic of this Vibrio genera is that the name Vibriate is derived from the characteristic vibratory mobility of this pathogen. Like when we keep the mobile in silent mode, sometimes a phone comes, then that mobile vibrates because of the vibration mode. Like that, this bacteria shows vibratory movement. So that's why the name vibrate is given to this Vibrio, is given to this pathogen Vibrio, because of the vibratory movement shown by this bacteria. These bacteria, they are also non-spore forming, means they are not producing the spores. They are also non-capsulated, means they are not showing the presence of capsule also. And these vibrios, they are mainly observed in marine environment and the surface water. So the most important member of this genus is the Vibrio cholerae, which is the causative agent of the cholera disease, which occurs in human beings. ह्युमन बीइंग्समध्ये होणारा जो कॉलरा डिसीज जो आहे त्याचा मेन जो पॉझिटिव्ह एजंट आहे तो आहे विब्रिओ कॉलरी. हा विब्रिओ कॉलरी हा जो बॅक्टेरिया आहे तो विब्रिओ जीनसचा मेंबर आहे. आता आपण जे कॅरेक्टर्स पाहिले ते विब्रिओ जीनसचे होते. कोणते कॅरेक्टर्स आहेत की हा जो बॅक्टेरिया हा जो ग्रुप आहे तो मेनली ग्राम निगेटिव्ह बॅक्टेरियाचा ग्रुप आहे ज्यांच्यामध्ये त्या बॅक्टेरियाची जी मॉर्फोलॉजी आहे कर्ड रॉड आहे. हा बॅक्टेरिया मोबाईल आहे, means shows the movement with the help of a polar flagella, and the movement which is shown by this group of bacteria, it is a vibratory type of movement. So the name vibrio is derived from this vibratory movement. Okay, this another characteristic which is shown by these members, that is they are non-spore forming, means they are not able to produce any kind of spores. They are also non-capsulated, means they are not producing any capsule. Now you have studied the bacterial ultrastructure. In that we have seen the outermost layer which is made up of polysaccharide that we call capsule. Such type of layer it is totally absent in this Vibrio genera. And these Vibrio bacteria, they are mostly observed in marine and surface water. Okay, and one of the most important member of this genus, that is the Vibrio cholerae, which is the causative agent of cholera disease in human beings. Human beings मध्ये जो cholera disease होतो त्याचा main causative agent हा Vibrio cholerae हा bacteria. Now this bacteria firstly isolated by the scientist Robert Koch from a cholera patient in Egypt, but he is not the first one who observed this, but the another scientist which is called as a Passini. He is the man who observed this bacteria in firstly in this patient. So the discoverer of this cholera bacteria he is the Filippo Passini. He is the person who firstly observed this bacteria up before the Robert Koch, and because of that the name of this bacteria is given as a Vibrio cholerae Passini after the name of this scientist. So firstly here this bacteria was observed by Filippo Passini. Later on it is also observed from the patient in Egypt by Robert Koch. Now first we will see the etiological characteristic of this bacteria, means their morphological, Gram's nature, biochemical characters, resistant characters, they are comes under this term etiology. So this Vibrio cholerae bacteria, they are short, curved, cylindrical rod which is measured about 1.5 micrometer to zero point 0.2 to 0.5 micrometer in size, and with rounded or slightly pointed, means at the end of this bacteria they are rod. So at the end they may be totally rounded or slightly pointed like a comb. Okay, this bacteria they also shows comma shaped structure. So because of that, earlier this bacteria was named as a Vibrio comma because of the comma shaped structure which is observed here. Now this curvature, comma shaped structure, it is only observed before isolation. After isolation on the medium, this curvature is lost. As we continue the subculturing, the curvature of this bacteria is lost. Sometimes this bacteria also shows S-shaped structure, spiral forms. Okay, it is because of the two or more bacterial cells they are lying end to end. Means they remain attached to one to end. Because of this, This shows S types of structure or spiral form, but this is not very common one. But the most common one is the comma-shaped structure, which is shown by this Vibrio cholerae bacteria. Now, one of the characteristic which is shown by this bacteria, it is that the pleomorphism. Pleomorphism means one more than one morphology shown by the bacteria. Here, this bacteria shows pleomorphism in old cultures. When this bacterial culture get old, it shows the pleomorphism character. First, suppose we firstly observe the fecal matter or that the mucus flakes from the acute cholera cases. We stain them and observe under the microscope. You are able to observe that these bacteria they are present in parallel rows like this. They are present in the parallel rows. So this type of arrangement which is shown by this bacteria, this arrangement Robert Koch called this arrangement as a fish in stream appearance. You can observe that lot of fish they flow in the water. They are present at the same line. So that's why it is known as a fish in stream structure. Now this Vibrio cholerae bacteria it is actively motile with a single sheathed polar flagella. Okay, because of the presence of single flagella, this bacteria is able to show the motility. And this motility which is shown by this bacteria it is of darting type. Okay, and you can observe this motility in this video. So this type of movement is shown by this bacteria because of the presence of a single flagella, and due to this it shows a darting type of mobility. When we observe acute cholera stool under under the microscope, you are able to observe actively motile vibrios which are indicated like a swarms of genets, like a whales which moves in the sea like that. or fish which moves in the sea. Like that, you are able to observe the motility of this fresh culture. Now this bacteria, when we stain this bacteria with aniline dyes, these bacteria, they are gram negative bacteria, and they are also non-acid fast. See here, you can observe here, this is the bacteria which is having a single polar flagella, and this is the gram nature of this bacteria. You are able to observe, this is the rod shaped bacteria. Here same is the photograph of the Vibrio cholerae bacteria, showing a bacteria with a single flagella. There is electron microscopic structure of a Vibrio cholerae bacteria, which is showing the curvature plus a single flagella. Now, next point in this etiology is the cultural characteristics. This Vibrio cholerae bacteria, it is strongly aerobic. Means to grow this bacteria, presence of air is must. Without air, the growth is very slow. Means when we try to grow this bacteria anaerobically, this bacteria grows slowly and their growth is very scanty. Means no growth observed there. The optimum temperature which is required for the growth of this pathogen Vibrio cholerae, it is a 37 degrees Celsius temperature. It is the optimum temperature which is required. But this bacteria can grow a temperature range between 16 to 40 degrees Celsius temperature. But the most required temperature for the growth of this bacteria is the 37 degrees Celsius temperature. Then next, this bacteria can grow very well in alkaline medium. Means in the presence of alkaline pH, this bacterial growth is very high. The optimum pH for the growth of this bacteria it is required is a 8.2, means it is alkaline pH. Above 7, that is alkaline pH. And the pH range in which this bacteria can grow, it is 6.4 to 9.6. This is the pH range in between this bacteria can grow, but the optimum growth or fast growth it is obtained when we provide the pH 8.2. Okay. Now another characteristic is that this bacteria can grow in the presence of a salt, that is sodium chloride. When we add 0.5 to 1% sodium chloride in the growing medium, the growth of this bacteria it is optimal. Now this bacteria can tolerate up to 6% of the sodium chloride in the medium. But above that, when we add 7% of sodium chloride, then it is inhibitory for the growth of Vibrio cholerae bacteria. Okay, so NaCl it is required for the optimum growth of this bacteria, and which is the how much is the requirement? 0.5 to 1%. It is the required for the growth of this bacteria. Now this bacteria it can grow very well on ordinary medium like nutrient agar. When we grow this bacteria on nutrient agar, after overnight growth, this bacteria produces moist, translucent, round disc about 1 to 2 mm diameter with a bluish tinge in a transmitted light. Okay, when we grow this bacteria nutrient agar, it produces this type of colony which is having moist. The nature of that colony it is moist, translucent means in light can easily transfer from the colonies of the bacteria. The shape of that colony is a round disc, and the size of this bacterial colonies it is 1 to 2 mm in diameter. And when we observe the transference of this bacteria or opacity of this colony of this bacteria on the nutrient agar medium, this shows bluish tinge when we observe the opacity. Now when we grow this bacteria on Macconki's agar, firstly the colonies which are produced they are colorless, but after some prolonged incubation that colonies become reddish in color because this bacteria ferments lactose very late. So that's why the colonies on this Macconki's agar after prolonged incubation becomes the reddish color. Here you can observe this. This is the bacteria which is present on the nutrient agar plate. Okay, you can observe round shaped colonies with a moist nature, and when we observe the opacity of this bacteria, you are clearly able to observe a bluish tinge in the colony. Okay, now this is the plate of McConkey's agar, where you are able to observe that there is a presence of reddish colored colonies because of the late lactose fermentation produced by this bacteria. Now, when we grow this bacteria on blood agar, the colonies, they are initially surrounded by a zone of greening, which later on becomes clear because of the hemodigestion. When we grow this bacteria on blood agar, firstly, around the colony there is a greenish zone is there, which zone, which is due to the killing of this RBC cells present in this blood agar, and later on this zone become clear because of the total digestion of blood in that blood agar. Now, when we grow this bacteria in gelatin state, they form various type of shapes. Infundibuliform shape, we can simply call this as a funnel shape. Napiform shape, turnip shape, we can call it as simply turnip shape. And this gelatin stab liquefaction is observed in 3 days at 21 degrees Celsius temperature. Now these are the various biochemical characteristics which is shown by this cholera bacteria. Now when we try to grow this bacteria on a peptone water, which is a simple medium, the growth of this bacteria is very fast. Within a 6 hours, you are able to observe fine surface pellicle on the test tube of this peptone water. When we try to break this pellicle by shaking, you are able to break this, and after that there is a turbidity and powdery deposit observed on continuous incubation of this bacteria in the peptone water. When you grow this bacteria in peptone water, within 6 hours, we are able to observe the growth of this bacteria in the form of surface pellicle. Okay, so this is another characteristic shown by this bacteria in a peptone water. Now, various medium are available for the cultivation of this Vibrio bacteria, and these are classified as follows. Here you can observe the gelatin stab, where this bacteria we have injected like by using a straight wire loop, and this shows the funnel shape. So this is a infundibuliform shape which is observed in gelatin stab. This is the growth of this cholera bacteria on the blood agars. Here, this is the clear zone around the colony of the bacteria. There is observed here. This is also a blood agar plate, which is where this bacteria is grown. That is a cholera bacteria is grown. This is the bacterial colony. Around that bacterial colony, there is a clear zone is observed. It is because of the digestion of RBC cells which are present there. Here, this is the peptone water. In that peptone water, we are grown this cholera bacteria. You can observe the surface pellicle, and this surface pellicle is produced within 6 hours. Within a 6-hour incubation period. 6 तासाच्या incubation period नंतर तुम्हाला लगेच इथे पेलिकल फॉर्म झालेला दिसून येईल. आता इथे बघा, टेस्ट ट्यूब उलटी केली आहे तरी ते मीडियम खाली येत नाही, कारण एवढं स्ट्रॉंग पेलिकल तयार झालेलं आहे. Now various mediums are available for the holding and transport. First holding and transport medium is the Venkatramana Ramakrishna medium. It is simply known as a VR medium. This medium contains crude sea salt, peptone water, and the pH of this VR medium, it is 8.6 to 8.8. As you know that this bacteria favors the alkaline pH, so this holding medium, it contains the same pH. Now this medium, it is added in bottles screw capped bottles near about 10 to 15 ml, and this medium, this VR medium, it is a holding medium. Why it is holding? Why holding mediums are required or transport mediums are required? Now, during the isolation of this bacteria or laboratory diagnosis of this bacteria, we require this holding mediums, because at some village place some person is suffering from this cholera, supposed to be suffering from the cholera infection, and we have to take the sample from that patient and bring it to the laboratory at some places which is present in the metro cities. So how to transfer that? So in order to transfer this sample from the patient to the nearest city, this holding mediums or transfer mediums they are useful. So this Venkatramana Ramakrishnan medium, this VR medium, it is used to transfer these samples from the village place to the city places for the laboratory diagnosis. Now in this Venkatramana Ramakrishnan medium, these Vibrios bacteria, they are not able to multiply, but they remain viable, live form for near about several weeks. Okay, in this VR medium, these bacteria remain live there for several weeks. So we can transfer this sample from villages to the nearest cities laboratories where laboratory diagnosis or identification purpose they can be carried out. The second transport medium, it is also known as a carrier layer medium. Now this carrier layer medium, it contains sodium chloride, sodium thioglycolate, disodium phosphate, calcium chloride, and it is also having alkaline pH 8.4. Now this is a suitable transport medium for Salmonella typhi, Shigella as well as Vibrio. This is a transport medium, which is a second transport medium we use for the transfer of the samples from one place to another. Then third transport medium, it is the autoclaved seawater. That is also means you know that seawater has a high pH because of the salts present there. So autoclaved seawater can be used as a holding medium. Okay, so these three mediums, they are available for the transport purpose or the holding purpose. Now another mediums are available for the enrichment of this bacteria. First medium, this for the enrichment purpose, is the alkaline peptone water, which is having pH 8.6. So this is the first enrichment medium. In this we can grow this bacteria for before isolation. We can enrich there in this medium. Second medium is the Monsour's tarocolate tellurate peptone water, which is having pH 9.2. So this is another enrichment medium which is used to grow this bacteria when this number of this bacteria is very low. So we have to multiply that bacteria fastly. So we enrich them by using these two medium, alkaline peptone water, then Monsour's tarocolate tellurate peptone water. These two mediums, they are good transport as well as enrichment medium. Here you can observe this is the VR medium, which is kept in the that is a screw cap test tubes, and here we have to add 1 to 3 ml of the fecal matter sample of that patient. This is the carrier layer medium, which is also present in a screw cap water. This is the alkaline peptone water medium, that is enrichment medium. Now different plating mediums are also available for the isolation purpose. Now up to now we have seen the transport as well as the holding medium. Now in next step we have to transfer this bacteria which is present in the enrichment medium or the holding medium into the plating medium. So different type of plating mediums are available. In that, first is the alkaline bile salt agar with a pH 8.2. This is a simple medium and it is longly used medium for the isolation of this Vibrio cholerae bacteria. And the morphology, the colony which is produced on this alkaline bile salt agar, they look like to that of nutrient agar plates where this bacteria can be grown. Now second medium plating media is the monsoons gelatin tarocolate trypticase gel agar medium. In simply we call it as a GTTA medium. This on this medium, when we inoculate the enriched sample on this GTTA medium and grow this bacteria for several or 24 hours, after 24 hours you are able to observe small translucent colonies with grayish black center and turbid hollow. This is the this is colony character which are shown by this bacteria on specifically this JTTA medium. What type of character is shown? This bacteria, Vibrio cholerae bacteria, produces small size colonies which are translucent, means light can be easily transferred, and the center of that colony it is grayish black center and turbid hollow. Around that bacteria there is a turbidity is there. Colony around that colony there turbidity is there, and the colony size of this bacteria it is 3 to 4 mm in size within a 48 hours on this GTTA medium. The next plating media it is the TCBS medium. This medium contains, why it is known as a TCBS? Because it contains thiosulfate, citrate, bile salt and sucrose. So because of that TCBS. Now this is the widely used commercial medium in laboratories for the isolation purpose. And when we grow this bacteria on this TCBS medium, this bacteria produces large yellow convex colonies which may become green on continued incubation period. As we continue growth of this bacteria on this medium, the colonies becomes green in color. You can observe here. This is the TCBS medium where this bacteria produces yellow convex colonies which are yellow in earlier. Later on it becomes greenish color. See here. Now this is the growth of the bacteria. We will call it bacteria on nutrient agar medium. This is the growth of this bacteria on GTTA medium. Okay, this is the black center around that turbid halo is there. Now this Vibrio cholera bacterial colonies they can be easily, suppose we have isolated the bacterial colonies on this medium. Now to confirm whether that isolated colonies they belong to the Vibrios or other bacteria, a simple test is there. That test is known as a string test. Now how to carry out this test? For this purpose we have to take a loopful growth of this bacteria from these selected medium bacterial colonies and mix that with a drop of 0.5% sodium deoxycholate in saline on a slide. If the test is positive, the suspension loses turbidity and becomes mucoid and forms a string when the loop is drawn slowly away from the suspension. You can observe this. So here you have observed that by a simple test we can easily determine whether the colonies which are produced on these selective mediums they belongs to this Vibrios cholerae or not by using this simple string test. Now the next etiological character, that is the biochemical characteristics. Thus this bacteria can ferment carbohydrate and produce acid but no gas. Now this is a simple test which is done practically in first year by using sugar media. This is the carbohydrate fermentation test where this bacteria where we use different type of sugars, glucose, fructose, or that is maltose, lactose, sucrose in that medium, and we test whether this bacteria produce acid or gas. So hello, म्यूट करा कोणाचा आवाज येतोय तो. Now the biochemical characteristics, this bacteria able to produce acid but no gas in carbohydrate fermentation metabolism. This bacteria can ferment easily glucose, mannitol, maltose, mannose and sucrose, but not inositol. Inositol, it is not fermented, it is not used by this bacteria, it is not used by the bacteria, also arabinols and lactose, but lactose it is used very slowly by this bacteria. Now in immune test, this bacteria produces indol, means indol, methyl red, August prosperous and citrate. Here this bacteria it is indol positive, and this bacteria is able to reduce nitrite. So these two characteristics, production of indol and reduction of nitrate to nitrite, these two properties they have been used for a cholera red reaction test. It is also a simple test which is used for the identification purpose of this Vibrio cholerae bacteria. Now that reaction it is known as a cholera red reaction test, which is tested by adding here we grow this bacteria in a peptone water. After the growth we are going to add few drops of concentrated sulfuric acid in that peptone water. If there is a presence of Vibrio cholerae bacteria, it produces reddish pink color. It is developed there. Reddish pink color developed there in this peptone water after the addition of concentrated sulfuric acid in that peptone water where we already grown this bacteria for near about 24 hours. And this pink color, reddish pink color, it is developed due to the production of nitrosoindol. It is a product which is produced by this bacteria in this peptone water. Production of indol and reduction of nitrate to nitrite. So the product it is nitrosoindol. It is produced there, and this nitrosoindol it produces pink color, reddish pink color after addition of concentrated sulfuric acid. Few drops of concentrated sulfuric acid in their peptone water. So this is the string test. You can observe there is a string is there where here that loop full of growth of bacteria is taken, 0.5% sodium deoxychelate is taken, and we mix them thoroughly, and we take a loop that bacteria and loop away, a string is developed. So this bacteria it shows that this bacteria belongs to the Vibrio cholerae. So this is the cholera reaction. If cholera bacteria is not there, no reddish pink color is there. So this is the positive test. This is the negative test. Now this bacteria is able to produce catalase enzyme and oxidase enzyme. So because of that, this bacteria it is positive for these two tests, catalase test and oxidase test. But it is negative for methyl red test and urease test. And this bacteria can decarboxylate lysine ornithine but not utilize arginine. Here this bacteria can liquefy gelatin, and this bacteria are able to produce various types of enzymes such as collagenase, elastase, chitinase, nucleotidase, decarboxylase, lipase, mucigenase, and amynase. These are the various enzymes which are produced by this bacteria while causing the infection. These enzymes are helpful for this bacteria. So this is the oxidase test. This production of this purple color dot here, it indicates the presence of oxidase enzyme in this bacteria. This is the negative test. This is the catalase test. You can observe bubbles formations there. Here we mix H2O2 and this bacterial colony, and after mixing, the bubbles are produced there. These bubbles indicate the production of O2, release of O2 molecule. Now, the resistance characteristic of this bacteria. This bacteria, Vibrio cholerae bacteria, they are susceptible to heat, drying, and acid. So, this cholera disease, it is mainly observed during the rainy season. While rainy season is there in our country, at that time, this cholera disease occurs mostly. So, because of that, you can observe here clearly that cholera vibrio, it is susceptible to heat. So during rainy seasons, we are told that use boiled water for drinking purpose. This is the reason. Because of heating, this bacteria can be easily killed. Because of drying, it can be easily killed. In acidic condition, it is also killed, but it is not killed in high alkaline condition. Okay, so you know that this bacteria favors the alkaline pH for their growth, and these bacteria, they can be easily destroyed by boiling near about 55 degrees Celsius temperature for 15 minutes. 15 minutes we can boil every item, then near about 55 degrees Celsius, this bacteria can be easily destroyed. On dried linen or thread, this bacteria may survive 1 to 3 days. Okay, on clothes, simply, or we can say that on clothes, this bacteria can easily survive for near about 1 to 3 days, and after that it dies. Okay, and on cover slip, this bacteria remain alive for 3 hours. Okay, now the survival of this bacteria in water, it is affected by various factors such as pH of that water. If pH is alkaline, it is favorable pH for this bacteria. It can grow very fast. If bacteria, the temperature of water is below 16 degrees Celsius, it can't grow there. Or if temperature of water is 37 degrees Celsius, it is helpful for this bacteria. Salt is required for the growth of this bacteria. If salt salinity is present there, it can favor the growth of this bacteria. So there are various factors which affect the growth of this bacteria in the water, such as pH, temperature, and salinity. In general, this even tor vibrio survives longer. Now there are two type of strains of this cholera bacteria are there, even tor vibrio and classical cholera vibrio. These are the two strains of this bacteria which are mostly affecting the human beings by causing this cholera disease. Now this even tor vibrio bacteria, they survive longer than classical vibrio bacteria. Okay, in laboratories, this vibrio survive for months in sterile seawater. And so in seawater, because of that, this bacteria can survive in seawater because of the presence of high salt concentration. So this we can say that this seawater, it is the favorable environment for the growth of this bacteria. It can remain there for various days. Now in grossly contaminated water such as Ganga water, this vibrio don't survive there for any length of time. The reason is that in this Ganga water, there is a presence of a vibriophage is there in Ganga water. So that's why this bacteria is not present in Ganga river water, because these vibriophages kill this vibrio bacteria by infecting. So that's why this cholera bacteria is not present in Ganga river water. Okay, and this bacteria can survive in clean tap water for 30 days, means our stored water. It can remain live there for 30 days. In untreated night soil, they may survive for several days in night, means below temperature is very low. In that condition, this bacteria can survive for several days. Now this bacteria, it is susceptible for common disinfectants which are used in our day-to-day life in our homes, like Dettol, Lysol. These are the common disinfectants which are used for cleaning purposes, and this bacteria, it is susceptible to this disinfectants. Also, when we clean the floor with using by this disinfectants, this bacteria can be easily removed from the floor. Now on fruit, this bacteria, they can survive for 1 to 5 days at room temperature. Okay, on room temperature, when we keep these fruits, they can survive for 1 to 5 days at room temperature. And when we keep these fruits in refrigerator, if that bacteria is present on the surface of these fruits, they remain for a week. So this is all about the